Welcome back to the deep dive. Today we are really going to get into the weeds of pharmaceutical safety testing,
We're dissecting the ICH S1C R2 guideline.
which is all about dose selection for carcinogenicity studies.
Exactly. So if you're a student or really anyone who needs to understand how we test new drugs for cancer risk in animals, this is essential stuff.
And our mission today is to make sense of the different ways to choose that all-important high dose for these long-term studies.
Right. Because how you pick that dose has been a huge point of contention for years.
A point of contention is putting it mildly. It was a source of serious international friction, wasn't it? We're talking about, the U.S. versus Europe and Japan.
Oh, absolutely. Before this guideline really harmonized things, you had this ideological divide. The U.S. FDA was, for a long time, almost exclusively reliant on one concept.
The maximally tolerated dose, or MTD.
The MTD. And they pursued it aggressively, sometimes, regardless of whether that dose really made biological sense.
And that sounds fine on the surface, right? Maximally tolerated. But it gets tricky with drugs that are actually very safe, the ones with low toxicity.
That's the core of the problem. If you just rigidly follow the MTD for a really low toxicity drug, you could end up giving a rat, say, 200 or even 300 times the equivalent human dose.
Which sounds super safe, but it's not that simple.
It's not. You end up with these massive doses that just overload the animal's metabolism, or they create weird nutritional problems. They alter the animal's entire physiology.
So the tumors you might see, they might have nothing to do with the drug itself.
Precisely. The guideline itself says the findings might not reflect what would occur following human exposure. You're not testing the drug anymore. You're testing the toxicology of a massive, kind of irrelevant overdose.
So it's that classic regulatory balance. You need a high dose to find a risk if one exists.
But that high dose has to stay relevant to the human situation. You want to maximize the safety margin, sure, but without creating a bunch of false positives just by, breaking the test system.
And that's what this guideline, especially the 2008 update, really tried to fix. It moved away from that rigid MTD-only approach.
Towards flexibility, towards a more scientific rationale.
Okay, before we dive into that flexibility, let's lock down the terminology, because MTD is still a really critical concept.
Absolutely.
When we say carcinogenicity study, we're talking about a long-term, usually 2-year study in rodents.
Right. And the doses for that long study are picked based on a much shorter one, typically a 90-day dose-ranging study.
And in that 90-day study, how do we define MTD?
So the MTD, the maximally tolerated dose, is the highest dose that's predicted to cause just a minimal toxic effect. But, and this is key, it still has to be compatible with the animals surviving in good health.
Is there a specific number or metric we're looking for, something a student absolutely has to know for an exam?
Yes, there is. The single most important metric for MTD is that it should cause no more than a 10% decrease in body weight gain compared to the control animals.
10%. Got it.
Right. And alongside that, you'd only want to see, minimal toxicity in organs and no major changes in their blood work that would mess up the study interpretation.
So MTD is all about observable physical harm.
It's a toxicity-based endpoint, yes.
Now, you mentioned the S1C R2 update and this idea of refinement under the 3 Rs. How did that change things in practice?
The biggest change was formally allowing a different approach, a pharmacokinetic one, to be used as an alternative to MTD, even for drugs that might be genotoxic.
Which means?
It means instead of pushing the dose higher and higher until the animals got physically sick, companies could cap the dose based on how much of the drug was actually in the animal system.
So it's based on exposure, not just toxicity.
Exactly. And that was a huge win for animal welfare. It directly reduced the potential for pain and discomfort from some of these artificially high MTDs.
Okay, so that perfectly sets up the core of the guideline, if MTD is one tool in the toolbox.
And the guideline gives you a whole toolkit. It officially lists 7 different endpoints you can use to select the high dose.
7. Okay, what are they?
You have toxicity-based endpoints, which is the MTD we just talked about.
Yeah.
Then pharmacokinetic endpoints, which is the big one, the AUC rule. Then there's saturation of absorption, pharmacodynamic endpoints, maximum feasible dose, the limit dose, and finally a catch-all for additional endpoints if you have a really strong scientific reason.
Let's focus on the second pillar then, the pharmacokinetic approach. You said administered dose in milligrams per kilogram can be misleading. Why is AUC a better metric?
I like to use an analogy here. Think of the administered dose, the MDKG, as how fast you pour water into a sink.
Okay.
Now, AUC, the area under the curve, is how much water is actually in the sink over time. The plumbing, the animal's metabolism, its excretion, is totally different in a rat versus a human.
So a rat might have a much faster drain.
Exactly. You could pour a ton of water in a high MDKG dose, but if the drain is super efficient, the sink never gets very full. The systemic exposure is low. AUC measures what's actually in the sink. It's what the body's tissues are truly seeing.
That makes perfect sense. So we're aiming for an exposure buffer, which brings us to what you called the big one, the most important number in this whole guideline.
It is. If you remember one number, it's this one. The critical value is a 25-fold exposure ratio.
25.
The high dose in your rodent study must result in a systemic exposure measured by AUC that is 25 times greater than the AUC in a human at their maximum recommended daily dose.
25 times the exposure seems like a massive safety margin. I mean, aren't we running into the same problem as before? Pushing doses so high that the findings aren't relevant?
That is a fantastic and fair question. And it gets to why that number was chosen. It wasn't just pulled out of a hat.
It's based on data.
It's based on a huge retrospective analysis. Regulators went back and looked at drugs we already knew were human carcinogens, things like IRC class 1 and 2A agents. And they found that testing at doses that hit at least that 25-fold exposure margin was enough to have detected those known risks.
So it's a historically validated safety net. It's proven to catch the really dangerous stuff.
That's the idea. It provides a very adequate margin of safety.
Okay, so if a company wants to use this 25x AUC approach, they need to have really solid data to back it up. What are the criteria?
There are a few key things. First, your rodent pharmacokinetic data, your AUC data, has to come from the exact same strain of animal and be given by the same route as in the main study.
No substitutions.
None. And second, you have to measure it for long enough to see if anything changes over time, like if the animal's body starts metabolizing the drug faster or slower.
And what about the metabolism itself? How it compares to humans?
That's crucial. You have to provide data showing that the metabolism is similar between the rodent and humans. If the human body breaks down the drug using pathway A, but the rat uses a completely different pathway B, your rat model is, well, it's basically useless for predicting human risk.
And there's one more tricky detail, one that I hear trips students up on exams. It has to do with protein binding.
Yes. This is definitely an advanced topic, but it's important. The general thinking is that it's the unbound drug in the blood that's biologically active.
The stuff that's free to interact with tissues.
Right. So if your drug binds really tightly to proteins in the blood, and if, say, humans have a much higher unbound fraction than rodents do, you can't just compare the total drug concentration.
You have to compare apples to apples.
You have to use the ratio of the unbound concentrations for that 25-fold comparison, the critical detail.
Okay, so we have MTD and the 25x AUC rule as the two main pillars. Let's touch on a couple of the others. What about the pharmacodynamic endpoint?
This is for very specific, highly potent drugs. Imagine a blood pressure medication. If you keep increasing the dose, you eventually reach a point where the animal's blood pressure drops so low it becomes life-threatening.
So the drug's intended effect becomes the limiting factor.
Exactly. That severe pharmacodynamic response becomes your high dose.
Yeah.
The only rule is you can't pick a dose so high that it invalidates the study, by causing all the animals to die from something other than cancer.
Right. And what about the maximum feasible dose, or MFD? That sounds less biological and more practical.
It's almost entirely logistical. If you're mixing the drug into the animal's food, there's a physical limit. You can only put so much powder in the chow before the animals either won't eat it or they get malnourished.
And that limit is?
It's generally considered to be 5% of the diet. But really, the goal of the whole guideline is to use things like the 25x AUC approach so you don't have to resort to these massive, sometimes less relevant MFD studies.
Okay, let's switch gears to what might be the most confusing part for students. Translating that 25x exposure ratio back into an actual physical dose, the limit dose.
This is where we connect the dots between the abstract idea of AUC and the concrete number of milligrams per kilogram.
And the official limit dose is?
1,500 mg/kg day. And this is considered the upper limit, but only if the maximum human clinical dose is 500 mg a day or less.
So why those specific numbers, 1,500 and 500? Where does that come from, and how does it connect back to 25?
It all comes down to a fundamental difference between rats and people, their metabolic rate. As we said, you can't just compare LG-QG directly. Scientists use a better method.
The body surface area adjustment.
The body surface area adjustment, exactly.
Hmm.
A tiny mouse has a huge surface area compared to its mass, so its metabolism is, running in overdrive compared to a human.
So you have to scale the doses differently to get a fair comparison.
Right. And when you do that math, that scaling, the 25-fold exposure margin from AUC ends up being equivalent to a much, much larger margin when you talk about the administered dose in mg/Ag.
Okay, so what is that equivalent margin? This feels like the key conversion factor.
It is. The 25-fold AUC ratio is considered roughly equivalent to a 150-fold mg/kg ratio over the human dose.
150, okay. So 25x exposure equals about 150x dose.
That's the conversion to remember.
All right, so let's walk through the math then. How does that get us to 1500?
Okay, so let's take a typical human dose, say 500 milligrams a day. For an average person, that's about 10 milligauge.
Right.
Now, to get the necessary safety margin, you need to give the rat 150 times that dose. So you take 10 milligrams and you multiply by 150.
And you get 1500 milligauge a day.
And that's it. That is why 1500 is the limit dose. It's not arbitrary. It's the dose needed to hit that 150x in milligauge margin, which is the equivalent of the 25x AUC exposure margin.
That clears it up completely. It's all about that surface area conversion.
Mm-hmm.
What's the biggest mistake or pitfall students need to avoid with this limit dose?
The biggest pitfall is just assuming that dosing at 1500 milligram automatically makes the study valid.
You still have to check the exposure.
You must check the exposure. The guideline is very clear. Even if you use the 1500 milligauge, you have to prove that the systemic exposure in your rodents is at least an order of magnitude, so 10 times greater than the human exposure. If it's not, the study might not be considered adequate.
We've talked a lot about the high dose, but the middle and low doses are just as important for making sense of the results, aren't they?
Oh, absolutely. They are essential for understanding the dose-response curve. They help you answer critical questions like, are the toxic effects we're seeing at the high dose just because we've saturated all the metabolic pathways?
If the drug is handled differently at the high dose than at lower doses.
Right. If the kinetics are nonlinear, it makes the high-dose findings much harder to interpret. So you have to pick those mid and low doses very carefully based on human exposure, the therapeutic dose, and all the mechanistic data you have.
Okay, this has been fantastic. Let's try and boil it all down. It's time for the essential takeaways. If you are learning this for an exam, what are the 5 points from ICH S1C R2 that you absolutely have to remember?
Number 1. The core principle. High-dose selection has moved beyond a single rigid method. It's flexible, it requires integrating all your data toxicity, PK, and PD.
Number 2, the MTD metric. The maximally tolerated dose is still a vital endpoint. It's defined by minimal toxicity, and that key number is no more than a 10% decrease in body weight gain in your 90-day study.
Number 3, the PK gold standard. The best way to measure systemic exposure is AUC, the area under the curve. It's far superior to just comparing doses in mg/kg.
Number 4, the regulatory ratio. This is the big one. Your high dose must achieve at least a 25-fold AUC ratio, comparing the rodent to the human exposure. That's your margin of safety.
And finally, number 5, the practical limit. The high dose is generally capped at the limit dose of 1500 mg/kg a day, assuming the human dose is moderate. And that number exists because it provides the necessary 150-fold mg/kg dose margin after you account for body surface area.
That really covers the fundamentals. So here's the final thought to leave you with. This guideline, S1C R2, is a really brilliant piece of regulatory science. It actively improved animal welfare by letting scientists use a more relevant exposure marker, AUC, instead of just an old-school toxicity marker like MTD. But even the document admits that these rodent studies have their limits. So the challenge for the next generation of scientists is this. If our best, most rigorous test, the 25-fold AUC rule, flags a drug as a carcinogen, but we know that finding only happens at exposures 25 times higher than any patient will ever see, what does that really mean for human risk? And how do we communicate that uncertainty? The conversation around cancer risk assessment is clearly far from over.